All right, so hi everyone, thank you guys for coming. I know that there's been a lot of questions on the Cam19 group. So this info session is with Dr. Mara Stone, who has a PhD in molecular virology, and her dissertation was in the development of HIV vaccines and virus evolution in hosts. Dr. Stone is currently a senior scientist at Core Labs at UCSF affiliated Vitalant Research Institute and an associate adjunct professor at UCSF Lab Medicine. Dr. Stone leads and has led multiple large international studies on infectious diseases and emerging infectious diseases, including pandemic responses. Dr. Stone is currently working on multiple COVID studies to understand the antibody response and persistence, mechanisms of vaccine production, as well as leading a very large national serosurveillance study to understand population level rates of infection. And my name is Dr. Aaron. I am a licensed acupuncturist here in San Jose, and so I'm also part of the Cam19 group. I was really curious about the new vaccines that are coming out in the next couple of weeks, and because Dr. Stone has such an extensive background in virology as well as vaccines, she agreed to help us kind of give us a little bit more knowledge. All right, so let's go ahead and start this little info session.
All right. Hello, everybody. My name is Mara Stone, and I'm a virologist, and I'm happy to present a very quickly put together kind of overview of some of the key questions that we were discussing earlier in the day, which led to sort of putting this slide presentation together, which I really am hoping that will address some key questions and help really combat a lot of the misinformation that's out there, but also provide a mechanism to stimulate further discussion that is based on science. Yeah, okay, so I'm going to address this, on a level that hopefully will really be very broadly accessible to people with a wide range of backgrounds and scientific sort of backgrounds and understanding. Okay, so we'll start with what the heck is a pathogen? So a pathogen is anything that's capable of causing disease in its host. It can be a number of different things. There's viruses, bacteria, fungi, protozoa, parasites, and even can be misfolded proteins or things like mad cow caused by prions. And that's sort of a technical term, and that's what I will use because I'm a scientist. So, and an antigen is part of that pathogen. So it's a small part, usually on the surface of a pathogen, that is recognized by the immune system and what triggers an immune response against that particular pathogen. So here's a little diagram here showing the pathogen and these differently shaped kind of proteins or antigens on the surface and the antibodies that recognize and bind to those very specific antigens. So a little bit of an overview about immunity and antibodies. So when a pathogen enters your body or you're infected by a pathogen, it starts to reproduce, and then your immune system, which is constantly on kind of surveillance mode, recognizes those pathogens as foreign and responds by making proteins called antibodies. So these antibodies are very large, so Y-shaped proteins that are produced by the mechanism of the immune response in order to identify a specific pathogen and try and neutralize it and prevent infection. So these antibodies stick to those unique molecules called what we just described, antigens that are on the surface of the pathogen, and it inactivates them by a number of different mechanisms, including just kind of binding to them and making them all clump together, or by sort of blocking the way that a virus binds to a cell and enters a cell. So essentially, it is preventing a particular pathogen from infecting a cell and being able to reproduce. For viruses, which we're going to talk about specifically today, a virus needs to get into a cell and kind of hijack the cellular mechanisms in order to be able to reproduce. So if a virus does not get into a cell, it can't reproduce and it can't cause infection and can't cause disease. So antibodies are a very critical part of the immune response. It does take a while for your immune response to learn how to do this. For pathogens it's never seen before. It takes 1 to 3 weeks, depending on what the virus is. So this is why you get sick the first time that you've ever been exposed to a particular pathogen. Is because it's taking a while for your body and your immune system to learn how to recognize and respond to a pathogen. So this is, so a little bit more detail on that. When a new pathogen and the specific antigens associated with that pathogen are introduced, it's not recognized. And again, it takes a while for those antibodies to be made to pathogens that's never seen before. But the next time that you get infected, your immune system knows exactly what to do and can do so right away. So immediately it ramps up your immune system to start producing the cells that produce those antibodies against that pathogen and prevents you from getting sick again. If you've already had it, then you've been exposed to it, your immune system knows what to do and can immediately respond. So you don't have to go through this, waiting period of 1 to several weeks while you actually get sick. It knows immediately how to respond and sort of cuts it off before you get sick again. So vaccination really is mimicking the process of going through a natural infection. So it's the same immunity that you would get from a natural infection, which means you actually got infection and you got the disease. But it has the benefit, of course, of the fact that you don't have to get sick in order to develop that immunity. Kind of gives your body a head start. So when it sees it, when you see the pathogen through exposure, it knows exactly what to do. So again, how do vaccines work? So they introduce a weak or inactive form of the pathogen, or in the case of like the Moderna and the Pfizer vaccine, an mRNA portion of the virus. And I'll talk a little bit about exactly what that means. And your body reacts by stimulating the immune system to create these antibodies. And this takes, a couple of weeks. And then the next time or not the next time, but when you're exposed to that particular pathogen, it knows exactly what to do and can defend against it if you get exposed to that virus. So it helps your immune system do the job it would do if you got exposed to the virus naturally, but it does it better and faster by really bypassing that time needed for your immune system to learn to produce those antibodies. Okay. I'll talk a little bit. Dogs are having problems again. And let's see. So a little bit about herd immunity. So this is important because vaccines don't just protect you, the individual. It also protects people around you. Because some people can't get a vaccine, like infants can't get all kinds of vaccines, or people with weak immune systems, like people who are going through ablative chemotherapy for cancer, or people with HIV or AIDS. They can't necessarily get vaccines, or there's other comorbidities that would be really recommended that people don't get a particular kind of vaccine. So how do those people get protected? One of the main mechanisms is by herd immunity. And that means that enough people in a population have immunity. Sorry, I think my kid is about to walk in the door and cause the dogs to start freaking out. So herd immunity means that enough people in a population are protected. So if a virus, a virus is infected. Sorry. So I have to do about that. 1 minute. Hey, come here. everyone. I'm giving a seminar here.
Sorry.
Say hi. Hi, this is my kid. All right, back to science. So herd immunity, it really means that if an infected person it comes into a population, that there's enough people around them that have a protective immune response that the virus is not going to be transmitted. So it's the people around them that are protecting the rest of the population from getting infected. You might ask the question, well, what is safer? Natural immunity, that is a result of getting infected and getting the disease, or getting the vaccine. Well, vaccines are generally much safer than actually getting the disease. I can probably safely say that nobody actually wants to get measles or chickenpox or COVID or smallpox or polio. And natural immunity happens when you get sick with the disease, but of course the disease can be very serious and detrimental. So the vaccine protects you as if you had gotten the disease, but you don't get sick from it. So really, herd immunity only works if most of the population is protected from infection, either by vaccination or having been infected. And again, that happens when most of the population has immunity. There's no way that a pathogen can spread. Really, it's a dead end for the virus to spread. So what the heck? COVID-19 vaccines happened really fast. And this is a very legitimate concern, and people are very right to question, well, what the heck? Like, how can we have this vaccine in just unprecedented time when we don't even have a flu vaccine that has worked with 100% efficacy? There's multiple reasons for this. And we have, when SARS and MERS were first emerging, there was a lot of effort developed to develop a vaccine. Hey, Rubes, can you grab the dogs?
Sorry.
Thanks.
Wait, sorry, what?
Can you grab the dogs?
Yeah.
Thank you. Sorry. So those have been worked on for some time, and SARS and MERS they're coronaviruses. They're novel coronaviruses. They're very similar antigenically or structurally to SARS-CoV-2. So we already had a very big head start in developing a COVID-19 vaccine. And I will also say that never before in history has there ever been this level of resources thrown at a problem. I cannot overstate, and I put this statement here, science was essentially put on hold with shelter in place. Labs were shut down unless doing work directly related to pandemic response. And thus, us scientists who are working on COVID were considered essential workers, because if you were not, you did not come to the lab. At UCSF and my lab, the institute shut down unless you were working on COVID. And so many people pivoted, just redirected their entire sort of lab and workflow to address many questions that needed to be answered on SARS-CoV-2, the virus itself, and the disease COVID-19. And additionally, there really was an unprecedented level of collaboration that was established. So the best minds in science, and I mean the best minds in science and the best labs, were all working together to a common goal. This has just never happened before. It's really how science should work. And as a scientist, I mean, it's amazing that all barriers came down. What is kind of not the best thing about science is that you are driven to publish, and you have to publish first. So you want to get your work out there so you can be a little bit secretive. Once it's out there, there's a lot of collaboration that happens, and that's an amazing thing. But here, nobody cared. Nobody cared who was first. Nobody cared about being the first to publish. Everybody just wanted to work together to try and address this pandemic. So again, there is legitimate concern about how the heck we could get this vaccine so fast and how do we know that it's safe. But for these reasons, I mean, billions and billions of dollars were put into making every available resource given to the scientific community to develop these vaccines. But, and then also say, what have I got on the next one? Oh, okay, so you might ask the question again, like why don't we not have a better flu vaccine? Why don't we have a vaccine for HIV? But also say, so vaccines come in many forms. They have very different genetic compositions depending on kind of the family of viruses. SARS-CoV-2 as a coronavirus is genetically very stable. It doesn't mutate. It's really kind of the same. There are small mutations, but it doesn't really affect the targets of a vaccine. So it's really easy to develop a vaccine for. We did it for SARS, SARS Classic and MERS. So as opposed to a flu virus, which is very different genetically, and its genome is segmented, and every year it kind of shuffles around a little bit. And the vaccines are produced with, a crystal ball, trying to anticipate the particular serotype of the flu virus that they anticipate will be circulating in the next year. And it's hard to get it exactly right because you're really just trying to guess. And so there's not complete protection, but it's usually a pretty good match. So you get at least partial immunity. And then with respect to HIV, very, very different sort of mechanism of reproduction and its genetic structure. Every time HIV mutates, every time it reproduces, it introduces point mutations. And it does this very, very fast. So it's like an entire population of different viruses, and it's sort of fast moving and very diverse. So it's very, very difficult to make a vaccine against a virus like HIV, as opposed to SARS-CoV-2. It's not a moving target. It's very stable, and we have very good technology to develop a vaccine against it.
So then, Dr. Stone, would you say that these two vaccines that come out would generally, obviously following the booster schedules, it would be basically like a one and done? Like it wouldn't need to be a yearly sort of thing because it doesn't mutate that often, or it doesn't mutate at all?
So that's an excellent question. So there's a couple of reasons why you might need to get a booster vaccine. One is because, or not even just a booster, but an additional vaccine. One is because it is genetically different. So the antigens that you're vaccinated against would be different, but also because the immunity that it elicits is not particularly long lasting. We do not know if this is going to be a one and done. And this is an excellent question, and I'm sure a question that's on a lot of people's minds. We have no idea. We do know that through natural immunity, antibodies wane, meaning they decline over time relatively quickly, which is a bit of a concern, really, that has significant implications both for establishment and maintenance of herd immunity, but also for long-term vaccine efficacy. So these are the studies that are really going to be ongoing so we can understand these questions and know how to how do we maintain lasting immunity. So we don't know. I suspect that there will need to be revaccination. We don't know what intervals, but that there will need to be revaccination in order to maintain that level of immunity. Okay. Yeah. So let's see. So I'll start, before I start talking about mRNA vaccines, I want to talk about what mRNA is. So mRNA is the body's way of reading the genetic code that is DNA. So if you think of DNA as a big instruction book, and that instruction book lives in the library, which is the nucleus of a cell. So every cell contains the entire instruction book for the entire organism, no matter what kind of cell it is. And you can imagine, like, you can't you can't check out this instruction book from the library. So you can't take the piece of DNA out into the rest of the cell so it can read and sort of carry out the functions based on the instructions in the DNA. But you can make a photocopy of bits of information. So mRNA is like a little photocopy of a piece of DNA that provides an instruction that is taken out of the library, so out of the nucleus, and then the mechanisms of the cell will read that piece of DNA in order to make protein. So we call this the central dogma in biology, which is information flows from DNA to make RNA in the nucleus, and that is sort of translocated outside of the nucleus into the rest of the cell, where the machinery reads that instruction and then makes proteins. And most of your body is made up of proteins. And this is where the instructions come from. So for mRNA vaccines, so these vaccines use sort of a man-made copy of the mRNA that would be produced by the virus in order to elicit an immune response. And that vaccine delivers this mRNA to the cells. And then the cells use the mRNA, as I was just describing, to build the protein, which is the antigen that an immune response is directed to, that would normally be produced by a pathogen. So this is a very small part of the virus, and they choose this particular small part. And in this case, it is the spike protein, because that is the protein that's on the outside of the virus, which will elicit the most protective kind of antibodies. So it's really the best target for those antibodies. These small parts of like the mRNA that produce the very small parts of the virus, which is the antigens, they're not capable of producing an entire virus. And therefore, they're really not able to infect you. Other types of viruses or other types of vaccines can also be delivering you small pieces of the virus or antigens, but also not a complete virus and also can't infect you. There are some vaccines that are inactivated, they're whole virus, but they're inactivated. So again, they can't replicate and reproduce and cause infection. There's some still other vaccines which are attenuated viruses, which means they're kind of engineered to replicate a little bit, but they do not have the capacity to sort of cause full-blown infection. All they do is replicate just enough such that your immune system can recognize it and then launch and establish protective immunity.
Okay, so another question too would be then, so basically this is that part of the central dogma that is to create the proteins. You know, you're inserting this mRNA so that it goes through translation to create those proteins specifically for the SARS-CoV-2 virus. So there's really no way, because mRNA is always outside of the nucleus, for it to actually affect human DNA. You're basically just telling these cells, okay, you're gonna build this protein that is part of SARS-CoV-2, not the full virus, just the one that's most recognized by your natural immune system anyways. And you're just bypassing that whole infection in general from the virus so that the body can naturally, if it does come into contact with SARS-CoV-2, it'll already have recognition of those spike proteins. Is that correct?
That's exactly right. Yes,
okay.
Exactly right. So that's very key. And I think I might even say a little bit more about that. But again, this flow of information, the central dogma, DNA to RNA to protein, it doesn't go backwards. If you introduced RNA, it cannot go backwards and somehow affect the DNA. Okay. And this is very important to clarify so that people really understand the science behind this and the mechanism of protection. And you know, it's this kind of information that will help people feel like they have a better understanding of the mechanism and can feel safe in getting a vaccine. So just a little bit more, what do I have here? Let's see. So yeah, just like all vaccines, the COVID-19 mRNA vaccines have been rigorously tested for safety before being authorized for use in the United States. So like they go through clinical trials. The phase 1 clinical trials involve tens of thousands of people. In the phase 1, they don't even look at efficacy. All they want to do is look at safety. So that's very key. So, and this technology of mRNA vaccination is kind of new, but it's not completely unknown. They have been, studied for use for some time. And again, the mRNA vaccines do not contain a live virus. It's just a little part of it. So there's no risk of causing infection or disease in the vaccinated person. So with that understanding, no virus is ever shed from any mRNA vaccination. So there's really no restrictions for post-vaccine contact. So if you're vaccinated, you're not going to be shedding virus, you're not an infectious risk, so you don't have to sort of quarantine because you might be producing virus that you're going to pass on to another person. And then again, to further address the question about what effect it can have on your genome, on your DNA. So the vaccine never enters the nucleus of the cell, so does not affect or interact with your DNA. There's some other benefits of mRNA vaccines in that the vaccine itself is not infectious. As I was describing, other types of vaccines, there's not very many sort of attenuated live vaccines. But in this case, there's no risk to actually getting infected by the vaccine. And also kind of bypasses a lot of the manufacturing criteria. So they can really be brought up very quickly. And they're composed of materials that are readily available in laboratories. That means you can standardize the process, scale it up very quickly, and the development part is faster than traditional methods, which would be, you're essentially kind of taking the mRNA and you'd be expressing that in some other organism, and that, or like actually yeast, or in eggs like flu vaccine. And then you'd have to sort of generate those protein subunits, and then you would take those subunits and then you'd vaccinate a person. So you're essentially bypassing that whole production process by putting it directly into your cells and letting your cells do the work of generating those antigens that will elicit immune responses.
Okay, that makes sense. I had one more question. Does this, and I'd have to read a lot of research, but does the spike protein that this mRNA vaccine create, does it cross-react with anything in our own bodies? Because I know that, SARS-CoV-2 has a very widespread effect on many systems within our body, circulatory system, nervous system, and that's why there's so many interesting long-haul symptoms. So when we get this vaccine and we're creating those spike proteins for the purpose of really being able to recognize if we do get infected with SARS-CoV-2, our body already recognizes it and basically shuts it down. Is that going to have any long-term, do we think there's going to be any long-term effects just from having the spike protein being expressed in our own cells or on the outside of our own cells?
Yeah, so that's a very good question. And there's many ways to address that. One is that when the mRNA gets in your cell and you express that protein, then your immune system recognizes it. The mRNA kind of goes away. It doesn't persist in the cell and keep pumping out antigen.
Okay.
So that's important. But then the spike protein itself, there's not really any analogous protein in the host, in people. So when this cross-reactivity, the concept of cross-reactivity has many implications. One implication it does not have is that it is similar to a human protein that you would be eliciting an immune response in a negative way.
Okay, great.
Okay, so let's talk about the Pfizer vaccine. This is also pertains to the Moderna vaccine as well. So this is this is sort of summarizing what already we're talking about, which is that the mRNA vaccines are giving the instructions for the cells to recognize the viruses. So again, the genetic sequence, the mRNA for spike protein is introduced into the cell, and then given the instructions to make that spike protein, and then this sort of synthetic mRNA is packaged in a lipid nanoparticle. I'll talk a little bit about that in the next slide, I think, which is just the delivery mechanism to get it into the cell. RNA is actually very easily degraded, sort of necessarily, if you think about how you have to regulate the expression of proteins in the cell. You have to sort of make the mRNA, make the protein, and then not have it hang around a lot, because sometimes you need to turn it on and off very quickly. But nonetheless, so those instructions are delivered to the cell, and then the cellular machinery makes the viral proteins that elicit the immune response. So the different components of mRNA vaccines, and they're very similar between the two, well, the big two, Pfizer and Moderna vaccines. So as I mentioned, lipid nanoparticles, which sounds kind of fancy, but really it is just like an envelope that encases the RNA to protect it. So these like kind of spheres that protect the mRNA and allow it to get inside of the cells. They're very, very small, about the same size as the virus itself. There is one particular lipid, this ALC 0315, it's positively charged to enable it to stick to the negatively charged RNA and kind of keeps the whole thing together. This is one component that can cause sort of side effects or allergic reactions. And I say, you know, side effects or allergic reactions, but those side effects generally are allergic reactions. In some cases, it can cause sort of a hyperimmune response, but that's very rare. Like, it's very rare. And I think I'll talk a little bit more about that in the next couple of slides. And then it has some other lipids like cholesterol, which exists in all of your cell membranes. And these also are like helpers that give structural integrity to these nanoparticles and sort of stops them from clumping together. It also contains salts, like ordinary table salts, and other inactive salts that make an inert saline solution that helps keep the pH of the vaccine close to that of physiological pH. It has some sugars in the formulation also, which act as a cryoprotectant. So it protects these nanoparticles when they're frozen, also protects them from sticking together. And then of course the vaccine is delivered in a saline solution, much like many other IV delivered drugs. And that is, so it matches the tonicity or the salt content in the blood. And then for preservatives, so the Pfizer vaccine, and I think also the Moderna vaccine, don't have any preservatives because it's a kind of a different mechanism of action. It kind of has these lipid nanoparticles that protect and stabilize the mRNA. So it doesn't need things like thimerosal, which incidentally have shown to be safe, but also legitimately raises the question of, are there other components of the vaccine that are potentially dangerous? But this is one thing that is not a concern, which is preservatives. Okay, so side effects. So most people don't have any serious side effects from vaccines. The most common side effects that people do get, for example, like soreness in the area of the injection, are usually very mild and go away on their own. Other kinds of side effects, you can get pain and swelling, you can get fever, chills, feeling tired, you can get headaches and kind of muscle aches. So really like general malaise and flu-like symptoms. And interestingly, a viral infection, most of the disease, or most of the discomfort you get from a viral infection, doesn't come from the virus itself. It comes from the immune response to that virus. So it's like your body working really hard to launch this attack on the virus. So when you get the flu, it's not the virus that is causing these things. It's your immune response against the virus that are giving you these flu-like symptoms. This is if you get sick after you get a vaccine, it's not because you've been infected. It is actually a very good sign that your body is building immunity against the disease. So again, these side effects are not that you're getting infected by the pathogen or the vaccine. And that's key. That's absolutely key. And I will say blanketly, unequivocally, without question, that these mRNA vaccines, you cannot get infected. There's no mechanism by which you can get infected by that pathogen by way of the vaccine. And then also, if you have previously been infected and you have natural immunity, that will not affect the safety or the efficacy of the vaccine. In fact, it will be helpful because you'll already have some immune response, and this will essentially act as a booster to that natural immunity.
One question. So I forget. So like you say in the very first slide, it takes usually 1 to 3 weeks to build natural immunity. With this vaccine, you know, obviously if I just get it, it's not like I'm going to throw my mask and just run around and like be around people. How long should we wait before we would be okay to not wear a mask, assuming, you know, that the vaccine is doing what it's supposed to do? You know, we're creating those spike proteins so that if we do come into contact with someone who was positive, you know, for sure we're not going to get infected or anything like that. What is the timeline for that?
Yeah, excellent question. So that's a great question. Just like if you got a natural infection, it would take 1 to 3 weeks. We do know by looking at people who have been naturally infected that you start to develop immune response within a week. So giving yourself enough time to launch an effective immune response would be like a week. I'd say to be very safe, give it 2 weeks.
Okay.
No one is saying that you can walk around without a mask.
Yeah, of course.
Just because you have protective immunity. Myself, I would, even if I had guaranteed 100% protection, protective immunity, I would certainly wear a mask because I think it's very important that people feel safe. Regardless of, I think it's very important that people feel safe, and I want people to feel comfortable around me. So I would wear a mask. But also the very few people, and really looking at the efficacy of these 2 vaccines, which is like 95%, there is some small fraction of people that don't get a very protective immune response. So better safe than sorry, I would say.
Okay.
Okay, what else? So other serious side effects. And really serious side effects from any vaccine are extremely rare. And so what does rare mean? Like how rare are we talking? So as an example, a typical sort of chances of a serious side effect would be 1 to 2 people in 1 million doses may have a severe allergic reaction. So, but that's not to say that it never happens. And I think just saying, there's no worry, just do it. That's just dismissive. And I think that's kind of problematic on the side of science and scientists, is that they're just dismissive because we're interested in population level efficacy. So just dismissing somebody's personal concerns about any side effects just really kind of shuts down the whole conversation about why vaccines are important, how they're safe, how they work. So, everybody can kind of be heard and, have that open conversation. So to say that it still is very, very rare. But in trying to understand what the effect of the vaccine can be in people who have comorbidities, it is true clinical trials are done in healthy individuals, and that's important because you have to first understand efficacy and mechanisms and correlates of protection before giving it to higher risk people. You necessarily have to do the clinical trials in healthy people, and then you kind of slowly introduce it into other populations to understand the additional risks that it poses to other populations. Sort of once it's approved for use, it is going to be rolled out to everybody. But if there is any question about whether somebody has a comorbidity or autoimmune conditions that might pose a significant risk, obviously that conversation is going to be had with the physician to weigh the benefits and risks of receiving the vaccine. So no one is saying that this is 100% safe for 100% of people. That's not true in any case, but this is, like anything else, a conversation that you would have with your physician to understand your personal risk. So what do those people do if they can't be vaccinated? This is again where herd immunity comes in. So those people that are not able to be vaccinated, that's not just COVID-19, but anything. And I'll also say that my daughter, when she was 3, she was diagnosed with cancer. She had leukemia. She had chemotherapy every day for 3 years, and she had no immune system. So in order for her to go to school, we had to rely on everybody in her class and in her school to have been vaccinated, because getting measles, getting chickenpox could have killed her. So I have very personal experience with this, that for some people who cannot receive a vaccine, they rely on other people to be protected, to prevent them from getting infected and getting sick. So that said, it is important to understand that getting vaccinated in any situation is much safer than getting the diseases that getting the vaccine that prevents a particular disease. Sure, you can get measles, you can get smallpox, you can get polio, you can get, anything, and you'll develop immunity, but that's unquestionably more dangerous than getting a vaccine. And on a population level, you cannot rely on everybody getting sick for just a myriad of reasons that we won't get into. That should be obvious.
Yeah, and I think even as healthcare practitioners, that's something that we're seeing, is that we're seeing a lot of people, they get sick, they get COVID-19, and then, they might recover, but then they're dealing with long-term symptoms. So it's like you're saying, okay, yeah, you got it, you have the antibodies for COVID-19, but if you could get a vaccine and not have those long-term symptoms, why wouldn't you, right? Like that's kind of the whole idea with this vaccine is so that you get, your body creates that spike protein that protects you from COVID-19, but you're not going to get the long-term symptoms that we're seeing with long haulers, right? That's kind of the whole idea.
Absolutely. Yeah, and we just don't understand why this is happening. We don't understand the mechanisms of pathogenesis, really. Like, we're just starting to understand these things. So it's true, and it's surprising, given it's a respiratory virus. It's surprising to the scientific community that it is having these really long-term clinical sequelae. So yes, getting vaccine is definitely safer than getting the disease. No question about it. Okay, so I think this is the last thing, which is yay, science. So really vaccines, and this is just general kind of overview stuff, they have reduced and in many cases just eliminated many diseases that killed or severely disabled people not that long ago. Nobody gets smallpox shots anymore because it doesn't exist, because it was by by establishment of herd immunity, it no longer exists in the population. Polio was once a very significant threat, causing death and paralysis across the country. Now, doesn't exist. Rubella vaccination, particularly in pregnant women, is no longer seen. The birth defects associated with rubella no longer seen because of vaccination. And so really the risk of vaccination without question is much, much lower than the risk of infection and disease. Nobody wants to say the risks are not zero. That the risks are zero. But it's important to know the facts. If you have questions, make sure that you're getting your information from reputable sources. And really, last kind of few thoughts, questioning science is good. Scientists do that. That's our job. Everybody should do that. But don't dismiss it out of hand because you don't understand it and don't have all the facts. And there's so much misinformation really being propagated now about SARS-CoV-2 and COVID-19 disease. And it's irresponsible and it's dangerous. Nobody doubts that not everything that you see on the internet is not true. Having the judgment to know what is and isn't true is difficult in many cases. So do be sure that any information that you get is from a reputable source. And conversely, scientists really do need to better address concerns for people who are questioning the safety and efficacy of vaccines, not just dismiss them, because we know empirically from the evidence that these are facts. So both sides being open to have that dialogue so we can understand both sides is very important. So I'm kind of looking at this as a very good opportunity to have that discussion. So that's what I got.
All right. Well, thank you so much. I think, this definitely lends even me for a lot of the safety and concerns in these coming up vaccines. So I'm excited to share this with the group. And if we have any questions, I guess we can reach out here and there. But this was all kind of everything that I think a lot of people wanted to know.
Good, great. And if you get additional questions, and do feel free to sort of circulate this presentation. If there are questions that come up that aren't addressed in this, I'm happy to kind of go in and revise to address them specifically, and even provide citations that people can go and read the primary literature themselves. There's some very good papers out there. A lot of them are obviously very technical, but some are quite accessible.
All right. Well, thank you so much, Dr. Stone. Have a good rest of your day, okay?
Thank you. All right, take care. Be safe.
All right, you too.